Good afternoon, all. Yesterday, we started discussing about DNA sequencing methods. Before that, we discussed about four major developments that enabled us to sequence the given fragment of DNA. We discussed about all those four developments. Then I also mentioned that there are two important methods of DNA sequencing. One is Maxam-Gilbert method, which is the chemical method of DNA sequencing. There we can label the DNA that is to be sequenced, and then different chemicals are used which will chemically modify the bases, and then these modified bases will be cut by using piperidine specifically in the DNA. So whatever the fragments produced will be run on the agarose gel, polyacrylamide gel electrophoresis in order to separate them. Because since they are radioactively labeled by autoradiography, we would be able to determine the sequence of the fragment. Then the next method of DNA sequencing is Sanger's dideoxy method of DNA sequencing. This is called dideoxy method of DNA sequencing because here DDNTPs. What are these DDNTPs? Dideoxynucleoside triphosphates are used as chain terminators. These dideoxynucleoside triphosphates, why they are chain terminators? Because at the third position of the deoxyribose sugar is no free hydroxyl group present. Since there is no free hydroxyl group present, the new nucleotide cannot be added to the growing chain of DNA. That's how whenever this DDNTP is added or incorporated during the synthesis of DNA, the further extension of the chain will not take place, right? So briefly, once again, I will discuss the Sanger's dideoxy method. Then we will go to the automated DNA sequencing. As it is mentioned, this was discovered by Sanger, who won Nobel Prize for his invention in the year 1980. And it also played very wide role in Human Genome Project. So here it shows the structure of DNTP, where we have the free 3' hydroxyl group at the third position of the deoxyribose sugar. Whereas in case of dideoxynucleoside triphosphate, we don't have the free hydroxyl group at the third position of the deoxyribose. That's how it acts as chain terminator. So this slide shows the difference between the structural difference between DNTP and DDNTP. This also we have discussed earlier. We carry out 4 parallel reactions here. Each one is specific for one chain terminator. All the 4 reactions, they contain the template strand, the fragment for which we need to determine the sequence, and a primer strand. As we all know, primer is required for the initiation of DNA synthesis. And for the reaction to take place, the enzyme that catalyzes the reaction is E. coli DNA polymerase 1 clenofragment. This clenofragment, it has 5' to 3' polymerase activity, 3' to 5' exonuclease activity. What it lacks is 5' to 3' exonuclease activity, which is very critical for this method. That is why all the 5' ends are going to be common. The 4 precursors which are used in each reaction are DGTP, DATP, DTTP, DCTP, and one of these precursors could be radioactively labeled. So this radioactive labeling can be done to any one of the precursors, or the primer what we are using can be radioactively labeled. In addition, each of the 4 reactions, so till here all the components which are present in all the 4 reactions are common. After this, the reaction mix is divided into 4 different parts, and in each reaction, specifically one chain terminator is added. So this diagram, it explains in detail the technique followed during Sanger's method of DNA sequencing. So here, if you see, this is the template for which we have to determine the sequence. So this is a single-stranded DNA of unknown sequence. And in this particular example, the primer used is radioactively labeled. This is radioactively labeled probe. If the primer is radioactively labeled, then the DNTPs need not be labeled. So after this primer is added, then the other requirements are DNA polymerase and all the 4 DNTPs, which are the precursors for DNA synthesis. After this, the reaction is divided into 4 parts. In each reaction, one specific chain terminator is added. So in this reaction, DDATP, DDCTP here, DDTTP here, DDGTP here. So when these 4 parallel reactions are carried out, first what we do is, what happens during this reaction that we can see and how the reaction products are formed. So here in one reaction, how the reaction products are formed, that is elaborated. Yesterday there was little confusion, how many G's, how many C's and all. Actually the point what I missed to explain is, here the primer is attaching. So leaving this portion, we have to consider the sequence from here. Leaving this portion, we have to consider the sequence from here. So in that case, when you use the chain terminator DDATP, where the DDATP possibly can get incorporated wherever there is T in the sequence. So how many T's are there in the sequence? From here you need to consider. So 1, 2, 3 positions, there is T present in the sequence. So that means there are 3 possible or potential termination sites in this sequence. So here, when C is there, obviously the G gets incorporated. That means that would be the normal precursor. Here, there is possibility for DDATP to get incorporated. Suppose if DDATP get incorporated here, then the chain termination takes place there itself. So what would be the product formed there in that case? In that case, the product formed would be this one would be the product formed. This product will be the product generated when the DDATP is incorporated at this position. If instead of DDATP, if DATP is incorporated at this position, first T, then further it gets extended. And here there is another possible termination site where DDATP could be incorporated. If DDATP is incorporated at second C, then this would be the product formed there, right? Instead of DDATP, if DATP is incorporated here, then further it will be extended. Then another possible termination site is this one. So the product formed will be this. I hope you are able to understand the point. We have the normal precursors DNTPs, and we have also incorporated the chain terminators DDNTPs. At all possible terminator sites, there is possibility for the normal precursor to be added. In that case, further extension will take place. Instead of normal precursor, if the chain terminator is incorporated, then and there the chain termination will take place. That's how all possible length of products will be formed. This is what I mentioned. A ladder of progressively longer molecules will be generated. And for this termination to take place at all possible termination sites, you have to take care of the reaction conditions in such a way that the incubation time for the reaction should be limited, one thing. And another thing is that the ratio of normal precursors and chain terminators should be maintained 100:1. 100 for 100 DNTP molecules, there should be 1 DDNTP. So in that case, you would see the termination at all possible termination sites, and you will have the generation of progressively longer, a ladder of progressively longer molecules will be generated. So once these reaction products are formed in all the 4 reactions, the reaction with DDATP, reaction with DDCTP, DDTTP and DDGTP. If you take DDCTP reaction, there is possibility that this DDCTP can get incorporated wherever there is G in the sequence. So here, how many G's are there? 1. Only 1. That means in this reaction, only 1 product will be formed. In the reaction of DDTTP, how many A's are there? 1. Right? So here also there will be only 1 fragment formed. In case of this reaction, DDGTP, let me see how many C's are there. 1, 2, 3. That means 3 fragments are formed. Once the fragments are formed, the reaction is complete, then you allow for the denaturation and then separate the fragments by polyacrylamide gel electrophoresis. So this is how we separate the fragments formed in each reaction. So each reaction product will be loaded in each line of the gel A, C, T, G, and then allow them to be separated by this polyacrylamide gel electrophoresis. And as you all know, the shortest fragment will migrate to the greatest distance, is it not? So, and each band indicates the chain termination site. That means, it indicates the base at which the chain termination takes place. That means here, and from the bottom of the gel, we have to read the sequence. So this indicates the last base to be C. And this band indicates G because it is in the line G. And this band indicates the termination at A. This band indicates again termination at A, G, T, C, A, G. And this will be the sequence of the longest nascent fragment. That means the longest DNA molecule that is generated in the reaction. In all the 4 reactions, this is the longest molecule formed in the reaction from bottom to top in 5' to 3' direction. The complementary of this sequence will be the sequence of the template for which we are trying to determine the sequence. So this is how the original gel will look like. This is hypothetical gel. Just for our understanding, it is drawn this way to get better understanding. But the natural gel will look something like this. I hope you are able to follow. If you have any questions, I'll give a minute. You can ask me the questions because this is very important slide. If you understand this, you understood the principle of Sanger's dideoxy method of DNA sequencing. Because even the next method, automated DNA sequencing, the principle is very same, just with one small difference, which made the entire procedure automated. So this slide, were you able to follow? Could you understand? If not, if you have any questions, you can ask me here. Any questions? If you are not asking any questions, then let me proceed further. Yeah, as we discussed in this technique, the 5' ends of all the fragments, they must remain at fixed nucleotide, and all the nascent chains and their lengths will be determined by the position of the DDNTP at the 3' end. That means that's the base where the chain termination took place. And correct ratio of DNTP and DDNTP is by maintaining 100:1, we can get the population of nascent chains terminating at all possible nucleotide positions. So that's about Sanger's dideoxy method of DNA sequencing. The next method is automated DNA sequencing. As I mentioned, this automated DNA sequencing also, the basic principle is Sanger's dideoxy method. The only difference between Sanger's dideoxy method and automated DNA sequencing is that for labeling, instead of using radioactive isotopes, in this technique, the fluorescent dye is used to label the nucleotides. And what is the advantage of using this fluorescent label as labeling material is that if you use radioactive isotopes, there are, because you all know radioactive isotopes are hazardous in nature. So when you use this radioactive isotopes for your experiments, there are certain protocols you need to follow for handling these radioactive isotopes. And once the experiment is over, you also need to follow certain protocols for the disposal of these radioactive isotopes. All this is very tedious. So as I mentioned, the fluorescent dye is not environmentally hazardous like our radioactive materials, and no special handling or disposal requirements are there when you use this fluorescent dyes as your label. And when we use this fluorescent label, Usually they are attached to DNTP, I mean DDNTPs, the chain terminators, whatever we use, we attach these fluorescent labels to these DDNTPs, unlike the radioactive isotopes. Radioactive isotopes are usually labeled to DNTPs, but here they are attached to DDNTPs, so that the chain terminated molecules, each one will carry a label at its 3' end. Obviously, each chain that gets terminated, because the DDNTP got incorporated in the at the end 3' end of the chain. Since the label is attached to DDNTP, that each fragment will carry the label at its 3' end. And here in this technique, a different fluorochrome is used for each of the 4 DDNTPs. Each DDNTP is labeled with a different fluorochrome. Since each DDNTP is labeled with a different fluorochrome, all the 4 reactions can be carried out in single tube. In Sanger's method, why we carried out 4 different reactions? Because we are using 4 different chain terminators. Since in this automated DNA sequencing, each terminator is labeled with different fluorochrome, all the 4 reactions can be carried out in a single tube. This is another advantage of automated DNA sequencing. That's how the whole technique could be automated. So this diagram explains how the automated DNA sequencing is carried out. So this is the double-stranded DNA for which we need to determine the sequence. We subject it to heat denaturation so that it gets, it is converted into single-stranded. Primer is required for the initiation to take place with its 3' free 3' hydroxyl group. And the other requirements of the reaction are, of course, polymerase enzyme for catalyzing the DNA polymerization reaction. And DNTPs are there, which are precursors for DNA synthesis. And here, for labeling, we are going to use 4 different fluorescent dyes. And these fluorescent dyes are attached to each of the chain terminators we are using in this reaction. Obviously, the 4 chain terminators are DD, TTP, DD, CTP, DD, GTP, DD, ATP. Each one is labeled with a different fluorochrome. Since each one is labeled with a different fluorochrome, we can carry out all the 4 reactions in the single tube. And after carrying out the reactions in single tube, and the remaining procedure is same. Since these chain terminators are added at all possible termination sites, the chain termination will take place and it produces a ladder of progressively longer molecules, which will be, so like this, all possible termination sites, the chain termination will take place and the ladder of progressively longer molecules will be generated. After these molecules are generated, we separate them by polyacrylamide gel electrophoresis, and then we can read the gel. Since the fluorescent labels are used, the whole procedure can be automated. In automated DNA sequencing, the reaction products are loaded into single well of the polyacrylamide gel, or we can also use the technique of capillary electrophoresis system. Once the products produced are separated by this system, then the gel can be run past the fluorescent detector, which will detect the fluorescence emitted by each band of the gel. The detector will identify the fluorescent signal emitted by each band, as I mentioned, and the detector also feeds the data to the computer. Then the computer will convert this data into the appropriate DNA sequence. Diagrammatically, it is very neatly represented here. The 4 DDNTPs, which we use as chain terminators, they are labeled with 4 different fluorochromes. Then, after the sequencing reactions are carried out, we can fractionate the products by polyacrylamide gel electrophoresis, right? So these are the population of fragments. Each fragment is attached with a fluorochrome labeled chain terminator at its 3' end. And after these bands are separated, we have a detector to detect the fluorescence signal emitted by these bands. Then the detector feeds the data into here imaging system in the sense the computer. The computer converts your data into appropriate sequence. So here each peak indicates a particular base. The color code is also there depending upon the fluorochromes we have used here. The color indicates the presence of base. So this is how it is converted into sequence. So the sequence is represented in this format when you determine the sequence by using automated DNA sequencer. Basic thing to understand here, since we are using the fluorochrome as dye, we can use 4 different fluorochromes to label 4 different chain terminators. That is how the whole reaction can be automated, because we can carry out the reaction in a single tube. The advantages of automated DNA sequencing: since it is automated equipment, it eliminates the need for radioactive isotopes to label. Since we are eliminating the radioactive isotopes, it reduces the volume of low-level radioactive waste generated in the campus. When there is no radioactive waste produced, we don't need to follow the cumbersome procedures like surveys, inventory, handling of this radioactive material wastage. And it also reduces the amount of manual DNA sequencing. The time could be saved because we don't have to perform autoradiography. If it is radioactive labeling, we have to detect it by autoradiography. We don't need to do that if we are using this automated DNA sequencing. As I mentioned, the work associated with radioactive waste is reduced, and it provides more reliable research results than the manual DNA sequencing. Thereby, we can maintain the integrity of the research because of the reliability of the results produced by automated DNA sequencing. Then we also have next generation sequencing. What is this next generation sequencing? Whatever the technique or improvement that is developed for this DNA sequencing after automated DNA sequencing is called next generation sequencing. Any improvement that happened after this automated DNA sequencing is named as next generation sequencing. This is shortly abbreviated as NGS, next generation sequencing. It's the new method for sequencing genome at very high speed and at low cost. So all the methods that are developed after automated DNA sequencing, they're all developed with the intention of producing the sequences at high speed and at low cost. This next generation sequencing is also known as second generation sequencing or massively parallel sequencing. Why is it called second generation sequencing? Because the automated DNA sequencing is considered to be first generation sequencing. So in that scenario, whatever comes next to the automated DNA sequencing is next generation sequencing or second generation sequencing. So using this NGS, an entire human genome can be sequenced within a single day. So that's the level of speed. As I mentioned, this automated Sanger method is considered to be first generation sequencing, and any technology developed after the first generation sequencing is known as next generation sequencing. This NGS that enables the sequencing of biological codes at a very rapid speed with low cost, and this is the main advantage over conventional method. So billions of short reads can be sequenced in one operation. That's the advantage. There are few examples of major platforms for this next generation sequencing. 454 from the company Roche, Solid by Applied Biosystems, Solexa is by Illumina. These are the major platforms available for next generation sequencing. So all these platforms, whatever mentioned, they vary in their principle, application and type of the data generated. But all these technologies have very common features that they generate sequence in a very high scale and very rapid speed, low cost, and DNA cloning is not required for this sequence to be performed. So these are the advantages in common for all new generation sequencing platforms that are available. So there are some pictures which will give the equipment of different next generation sequences from Roche. This is the example 454, GS-FLX 454, HiSec 2000 from Illumina, 5500 XL Solid from ABI, GS Junior, Ion Torrent. These are few examples of this next generation sequences. So this is all I wanted to discuss in this DNA sequencing methods. Just to quickly summarize, we discussed chemical methods and enzymatic methods. In chemical method, Maxam Gilbert method, where specific chemicals are used for modification of bases. After modification, piperidine is used to cut the DNA backbone of the, so sugar phosphate backbone of the DNA. Then the fragments generated will be separated by polyacrylamide gel electrophoresis, and it can be read from the bottom of the gel. Whereas the next method, enzymatic method, we have the basic Sanger's dideoxy method, where chain terminators are used, by which a ladder of progressively longer molecules produced. These molecules will be separated by again electrophoresis, and from the bottom of the gel, when you read the sequence, that gives the longest nascent DNA molecule produced, and the complementary of that sequence will give the template strand DNA sequence. So in the Sanger's dideoxy method, the radioactive labeling is used. Sorry. Then the next method is automated DNA sequencing. The principle is same as Sanger's dideoxy method, but the difference is here the labeling is done by using fluorescent dyes. Here labeling is, the labels are attached to the chain terminators, the 4 DD-NTPs. For 4 DD-NTPs, 4 different fluorochromes are used. That is how we can automate the technique by carrying out all the 4 reactions in one tube. So after the fragments are produced by chain termination in this automated DNA sequencing, they can be separated again by page. And since the labeling is done by using fluorescent dyes, the signals can be read by the detector. The detector feeds the data to the imaging system or computer, which converts this information into the appropriate sequence. So it has several advantages, like you don't have to handle the radioactive isotopes, because of which we can reduce the amount of task involved in the experiment. Comparatively safer, and it is very fast since the technique could be automated. Then we also have next generation sequencing. Whatever the technology developed after automated DNA sequencing is next generation sequencing. The main principle for developing this next generation sequencing is to produce the sequences in a very high speed and low cost. Several examples are given for next generation sequences. Thank you so much. If you have any questions, you can ask me.